Thank you so much to all of our speakers. Much appreciated for all your contributions. So now what I would like to do is welcome our speakers. If they're able to, please do turn on your microphones. And if you would like to and are able to, please do turn on your cameras as well. And now we will be having a roundtable discussion. So what we would like to do is invite you to take part in the Q&A session. So thank you again to all the panelists. Your talks today have really clarified interesting opportunities and trends in the cancer-related pharmaceutical business, and it's provided an understanding of quite interesting development opportunities as well. So following on from that, we'll begin asking our experts some deeper questions on some of the subject matter that you discussed earlier, and hope to respond to some of the questions that have been submitted in chat as well. I have a couple of questions which I'd like to start off with if possible. And by all means, please do answer. It's open to all of you. We do invite conversation. So, you know, if one person wants to answer and if another wants to add to that, or perhaps wants to have a different sort of response opposing that, then that's absolutely fine as well. So to kick off, I'm quite interested from the perspective of technology, what technologies actually caused such a rapid vaccine development, so such rapid cancer vaccine development. It'd be interesting to hear about those technologies and what actually drove that as well.
I think there have been a couple of developments, which I also kind of highlighted, I think, in my presentation. So I think firstly, people started to realize that cancer vaccines on their own are probably not efficient enough to actually treat the cancer in the majority of cases. So you will need to be think about combining the cancer vaccine with something else. Well, we didn't have a lot available to us, of course, in the early 2000s. So now we have the checkpoint blockers, and they form a very nice combination therapy there. But the other thing is that we really start to appreciate that that you really need to get a certain quantity of T cells and also quality of T cells to actually drive an anti-tumor response. So quantity meaning, if you, for example, look at some of the antiviral vaccines and the COVID vaccines, and if you look in patients there, actually a quite substantial amount of circulating T cells of these patients are directed against the COVID vaccine. So very strong immune response. And you don't see that a lot in the cancer vaccines. So you really need to have an immune response where at least more than 1% of circulating T cells is directed against the antigen of the cancer vaccine. And that T cell should be both a CD4 and a CD8 T cell. That's very important, because then you actually get the most efficient CD8 T cell activation and also the memory response. The memory response is also a very important one. So I think those are probably the key points. And of course, now we also have the technology, right, to kind of identify those patients that can actually respond to treatment. We have the cost of DNA sequencing, RNA sequencing have come down quite a bit. So now you can start to use all kinds of parameters to select patients.
I may comment this question. I think science is the reason for this. Currently, we have a big direction in science known as the omics sciences. And omics sciences can be considered as a high throughput technologies. And they appeared in science many years ago. And after that, we can able to analyze in numerous molecules in single run. And this is very important. For example, we can take new antigens just to find genes, highly mutated genes. We should apply genomics technologies. This is very important. It means that technologies we usually use in science, we are going to apply in biotech. If we have genes highly mutated, we should know that we have mRNA. It means that we should apply transcriptomics. But if we have mRNA, it doesn't mean that we have protein expressed from a gene. This is a big problem. There is not correlation between mRNA and protein. So to find protein, we should apply proteomics. It means that we should apply high throughput technologies, and that is why I see that we have a rise of technologies connected with anti-cancer vaccination. All the proteomics can be used to find tumor-associated antigens in high throughput way. Moreover, you can realize from my presentation, antigenic assay technology is based on proteomics. Proteomics is high throughput analysis of proteins. So we're able to generate a big data in different levels, and we can analyze them by applying bioinformatic treatment. For example, artificial intelligence can be applied. Now, that is why I think all is about science, and what we use in science, it will be applied in biotech just several years later. That's all. Thank you.
So just following on from that as well, and I know this was touched upon earlier, with regards to the technological challenges, why did so many trials fail in the past?
Maybe I can comment this question. Because we haven't principal solutions. If we are going to use tumor-associated antigens, it's definitely we will deal with tumor escape. And I don't see solution for this. CAR-T technology can solve some problems connected with MHC restriction. That's all. That is why CAR-T succeeded, but only for particular cancers, only for particular. And I think this is a real exception than rule. CAR-T technology doesn't work for solid tumors. I think we have same situation with new antigens. We can improve different parts connected with development of vaccine based on new antigens. But I don't see a strong base to succeed in this direction, because trying to use highly mutated antigens in order to produce stable vaccine, very questionable. I don't think so. And we know that tumor is heterogenic, and if you take a sample from tumor to prepare vaccine based on new antigens, it doesn't mean that if you take sample from other side of tumor, vaccine will be same. Only if tumor is one clone generated from one clone of tumor cells, you may expect that new antigen vaccine can work. In other case, you will destroy a part of tumor, and you hope that this effect will be evident. That's all. But other part of tumor will be able to grow more rapidly. The empty place will be occupied by other cells. So there are no principal solutions for this. What we can do? We should have a very simple and very straight approach to solve main problems. For example, antigenic essence technology may be considered as such approach. I think we proposed a very good solution and principal solution based on main principle of vaccination and removing all shortcomings.
Okay. So just putting this back out onto the floor, how would that relate to, and I think one of the questions we had earlier asks about this as well, to the more rare cancers, and sort of spinning off from that question, I believe it was asked about whether we should be looking at personalizing these vaccines for those that are affected by the more rare. I'll just double check who I think asked that question. I think it might have been Robin Privet for the rarest tumor types. I would think you absolutely need to personalize vaccine as well. So while your response, Piotr, makes complete sense, how would that relate directly to those lesser known types that aren't as easy to combat with those methods?
Oh, pardon, can you repeat the question?
So with what you've said, we talked about the challenges, and we talked about the failures in the past. And it seems it's a great, your response was sort of, you know, one rule for implementation overall for the rollout and development of vaccines. But how would that work with the rare vaccines? How would you engineer that, and how would you be able to tailor? And I think one of the comments from Robin Privet earlier on was that for the rarest types, you need to personalize your vaccines. And when you're talking about personalization, the kind of mass formula doesn't necessarily apply.
Okay. Okay.
And that's open to everybody as well to answer.
Okay, I can answer this question. I think if we have very straight approach, we shouldn't deal with personalized vaccination at all. Maybe it's my wrong, and I didn't clarify it in my presentation. I said that we are going to use primary culture, and someone can think that this is a personalized approach. No, this is not a personalized approach. Primary culture relates to one batch of our product. We should use primary culture in order to prepare vaccine suitable for targeting of microvasculature and tumor vessels. We can read cell surface. That is why we know we can't use immortalized cells. That's all. That is why we're going to use primary culture, and we believe that we will succeed. That is why we are going to vaccinate numerous people. We are going to produce a relatively cheap the vaccine. So personalized approach is very complex, very expensive, and mainly relate to new antigens. This is not our approach. Maybe in future we can produce. We're able to do this. Our technology suits for this, but I think we should start with vaccine which can be produced for all people, and maybe in far future, personal approach will be also applied. So my personal opinion, we shouldn't make many efforts in direction of personal vaccines, so and new antigens too.
Of course.
Sorry.
So one of the comments actually from an attendee who remained anonymous was that his daughter, his or her daughter, apologies, was actually getting a personalized vaccine due in December to keep her in remission, and it has taken 6 months to analyze and produce. So obviously, from what you've said, there is a case for it, but it's a longer process that, you know, a lot of...
Yeah.
Okay. Just about time. Just information about time. To produce, for example, our antigenic essence, 3 months is required. But if we're going to about personalization, we have personalization in case of dendritic cell-based vaccines. We are going to use allogenic antigens, but dendritic cells are going to use autologic. And in this case, if we have already prepared antigens, we can produce dendritic cell-based vaccine within 1 week, because we should just isolate dendritic cells and load with antigens, and some portion will be stored, but after 1 week we can already vaccinate.
So there's a scale. Thank you, Piotr. Dr. Goches, would you like to weigh in on that as well? Or
yeah, I think, you know, it's an interesting question, of course, if you can go for if you can make a personalized vaccine for a rare tumor. There's not a full answer to that. I mean, it really depends on the tumor. If it's a tumor that has a very low amount of new antigens, then it's very difficult to make a personalized vaccine, right? So then you need to come with a vaccine targeting, for example, a tumor-associated antigen or a cocktail of that. And also,
let's use, for example, like I'm trying to think of, say, SRCC or something similar along those lines. Does that apply to this case here?
I
think just as an example, I think it was signet ring cell carcinoma as an example. It's quite rare, difficult to detect, but very, very unique to the individual.
Yeah, it's hard to say. I mean, you really have to sequence the tumors and see if you have a suitable number of new antigens in there. And then, like Peter explained, you have to be able to predict which of those are actually immunogenic, are presented on the surface of an APC. And also that's also processed in the correct way by the MSC class molecules. And then you actually have to make a product, infuse it back to the patient. And probably you need many new antigens. So if you don't have that in the tumor, then the neoantigen approach won't work. So then you need to start to think about different approaches, such as the tumor-associated antigens that were also covered. That can still work, of course. But for the vaccine to work, you need to also think about activating the APC, right, with an adjuvant, or combining with PD-1. And that really depends also on the tumor microenvironment status, right? If there are no T-cell infiltrates in there, it's going to be difficult to get a response with PD-1. Having said that, for example, glioblastoma, that's a very poor outcome brain tumor. That's considered a cold tumor, but for some reason, people have been able to actually trigger immunogenicity with tumor-associated vaccines in that tumor. That was quite a surprise to the field because these tumors don't contain any T cells, or very hardly any T cells. So sometimes cancers have surprises there. It's really you really have to look then in the patient sample. So sequence it, and then you can get a sense of if it works or not, if it will be suitable for a cancer vaccine.
I think there's a common theme with some of the questions and comments that we've had in chat is, obviously a lot of people would like the idea of personalization, but appreciate that you can't necessarily mass produce that. And it might not be as effective in terms of the time scale to be able to make that much of an impact. So just moving on to some of the questions in the Q&A, I feel as though I have to respond or ask a question posed by Jose Sanchez, who should have really been on our panel. You've got some very, very interesting questions there. So we can choose, perhaps, if we look at one of your earlier questions. So could anti-cancer viruses be replaced by CRISPR CRISPR vaccines, and can cancer cells be mutated back to healthy cells?
No comments.
That's quite a specific question.
This is related to editing of genome, how it is connected with cancer vaccine. Maybe, but I don't know.
No problem. Well, perhaps we should go back to the concept of technology. So another question from Jose is, AI pattern recognition should have output neurons that recognize patterns, but also it must have an additional output neuron that indicates probability of unrecognized pattern for a certain region. Does the AI have that neuron? Such output neuron leaves a door to spot new patterns instead of delivering bogus output. It's a question for researchers to the AI company. I suppose it's posing a thought, but also asking the question, whether AI has that neuron.
I definitely think there's AIs that are in development, that if you are scanning a wide set of known protocols and profiles in a particular sample set, if there's something that's outlying, you can certainly have it focus on what's left over, as opposed to just what you're looking for on those known ones, so that you'll have what's unrecognized and then be able to work on it from that other end. But you'd have to exclude the items that you know are already existing before you can today find the things that are the outliers. So you have to step into it kind of from a backwards scenario.
Okay, I may say several words about neural networks and the application in biotech and in science. I think artificial intelligence can be easily applied for some fields of science, of biotech. For example, to find new composition of drugs, mainly low molecular compositions. And we know such approach known as QSAR. It means quantitative structure activity relationship. And in this area, artificial intelligence can be applied with big success. Currently, it's very difficult to apply artificial intelligence to big data. This is a big problem. We can see a huge number of scientific papers because we have numerous bioinformatists and we have numerous databases with freely available big data related to different type of data, proteins, low molecular substances, genes, and we have a numerous application of artificial intelligence, but we will not see a very successful examples in industry because application of artificial intelligence to big data, to real big data, make situation more complex. Things become worse. But maybe 10 years later, I think this will be a real trend. Now this is a real trend in design of low molecular drugs and in science. But 10 years later, we will see how artificial intelligence will be applied in the industry.
Perhaps even sooner. A question for Natalia and potentially Andre as well, referring to how cancer vaccines overcome the challenges that tumor antigens rapidly mutate to evade the immune response. So just to repeat that, how will cancer vaccines overcome the challenge that tumor antigens rapidly mutate to evade the immune response?
Yeah, I think I can start with this question. So first of all, as we discussed, one of the main approaches is using multiple antigens in one cancer vaccine. So if, for example, we're having just few neoantigens in constant, it means that if we are constantly targeting only these few neoantigens, at some moment of time, there will be some cancer cells group which will develop the mutation and start a new clone or like a new tumor which is resistant to this treatment. But if we're targeting multiple, like dozens or hundreds of antigens at the same time, tumor doesn't really have this chance to escape that easily. And at the same time, also we might target not only cancer cells which are mutating really fast, but vasculature of the tumor, like microenvironment. And by microenvironment, I mean the environment around the cancer cells, which includes vessels, fibroblasts, signaling molecules, and other factors. So I would say that these two are main approaches how we avoid this tumor escape because of the rapid mutation rate.
Thank you. Thank you. I
So
think Natalia is talking about Santavac. We are going to target vasculature and we are going to use the diversity of antigens. This is a way how we can manage mutations in cancer. Thank you very much.
Dr. Gershis, would you like to weigh in on that question as well?
Yeah, I think Natalia was quite right there. I think another approach is more on the clinical development strategy. So the earlier you give the vaccine to patients, so the early in disease process, the higher chances that you have that the tumor is not so heterogeneous. So you have a bigger chance of actually getting good results with your cancer vaccine. So that's why a lot of people are now interested in actually applying cancer vaccines together with PD-1 in the adjuvant setting, because the tumor has been debulked by surgery or chemotherapy. So there are only some residual cancer cells there. So if you then apply the cancer vaccine, then the tumor doesn't have a lot of chance to escape. But of course, then you still need to find a way to actually get your vaccine very early in the treatment line. But the good thing is that most cancer vaccines are actually pretty safe. So that's an advantage.
And what about the political landscape? Obviously, this will vary in various different states and countries. What's involved with that process, with all the various regulators that exist and have their own operating rules? How would that work with obviously a lot of what's been discussed and getting things approved, and particularly when we're talking about vaccines?
Yeah, it's a good question. Obviously, for any vaccine product, you first always have to do your phase 1 trials to show that it's safe. And if you have an excellent safety profile, it definitely helps you to position your products already early in the treatment lines, like in the adjuvant setting. Still, you will be, I guess, a little bit dependent on clinical key opinion leaders trying that, I would say. Of course, also you need to have consent of the regulators. But because normally with a cancer product, indeed, you have to start with very advanced stage patients that progress to later treatment lines. That's always a problem. But if your product is really safe, showing some good immunogenic responses in patients, so a high degree of circulating T cells, good safety there, then you might be able to convince some key opinion leaders to actually try it in combination with PD-1 or a different checkpoint inhibitor. I mean, there are several others emerging now in your target indication.
Thank you. And just one of the questions that was asked earlier, and I think we may have answered this as part of your presentations, but just to touch upon it again, in terms of the success rate of such anti-cancer vaccines as of now.
At the moment, it's pretty bad, unfortunately. I mean, I think that the Pro-French and the BCG bacterial products are probably the only cancer vaccines products are on the market right now. And so far, almost all clinical trials have failed for reasons that there was simply not sufficient immune response in patients, right? Clearly, so there are a lot of companies now in the clinic with their products. So I guess we'll just have to wait for the randomized clinical trials to read out. So it are exciting times, and I think the field is kind of looking forward to get a success there, right? Yeah.
So in terms of those success stories, actually, Dr. Andrei Bavelo, if you're available to answer, what would you say are the most notable success stories in terms of biotech rounds and IPOs recently?
I mean, in the cancer vaccine space, probably first that comes into my mind is BioNTech and Moderna. They have diversified portfolios, but also include some cancer vaccine products, as we have seen in the presentation, in my presentation. So they were both quite successful with their APOs, especially Moderna. And I believe one of the reasons they were so successful is because they were focused on many things, and also the money that they raised basically led them now develop these products as well, and I believe they will get some success. And also, I mean, Sensei Biotherapeutics is the company that raised 133 million, if I'm not mistaken, quite recently, and so they have also the product in the pipeline. But again, the key metrics right now about this industry is not it's not about IPOs so far, because we are in the proof of concept stage right now. And as I have seen on the graph, we will see either very skyrocketing growth, or if many failures occur, we potentially might might be quite disillusioned and quite disappointed. But from our analysis, we do believe that due to new technologies coming into play, both on delivery side and on the antigen selection side, we do see some hope that next year, maybe in the next 3 years, we will see some preclinical success stories and probably clinical success stories. And of course, going from 0 to 1 will be an important part in this process. And but finally, my last point is that in this game, I would say the most important item for a successful company to have the platform rather than just a single set. The platform-based business will win here because in the cancer space, it's important to pivot easily between certain indications. And I would say the platform-based businesses here will certainly be heads ahead, like mRNA, for example, or antigenic essence or some other technologies like that, because they can simply pivot quite easily, relatively easily, into certain cancer types, certain patient subgroups, etc. So that's important.
And finally, I think, apologies, I know we've run over, but if I may just ask one last question, which can be put out there, and I think Dr. Gertjes, you may be first to answer this as well, which is the critical bottlenecks for startups, so biotech startups. What are the critical bottlenecks that they would be facing, especially in the initial phases of company development? And what advice you would have? I think we do have people that have joined us today who could be from such companies.
Yeah.
This is out to the floor.
Yeah, so it's definitely not an easy time for a cancer vaccine developer to be in, for reasons that the presenters have shown. There's a lot of competition, people using different types of antigens, different types of platforms. So it makes it very hard for investors and also pharma to evaluate those propositions. And also immuno-oncology in general is a very competitive space. Cancer vaccines are probably a very nice combination partner with anti-PD-1, but almost everyone in the world is trying to combine their drug with anti-PD-1, and that does not necessarily have to be a cancer vaccine. So you're not only competing against the cancer vaccines, but you're also competing against different treatments. And in the end, what counts is, of course, that you are able to transform the standard of care of a certain target indication. So I think as a company, what's very important there is that you start to look into the indications where your products would fit very well, make a couple of target product profiles, and where you highlight kind of where you're going to, what will the product label of your cancer vaccine, what will be the combination product, how will you select the patients, those kinds of things. And be prepared that the landscape will shift a little bit over time when you ultimately get to the market. I think you need to be preparing a pretty strong preclinical data package these days. So some of the things that are already presented, so showing basically all the hallmarks of the cancer immunity cycle in preclinical animal models, good efficacy there, and the memory responses is very important, I think. And maybe as a nice to have to actually bring some human ex vivo data there as well. I don't see a lot of companies doing that, but it definitely de-risks the company from my perspective. Because, yeah, I mean, in the end, animal models are not so predictive for the humans. So there's only some, I mean, they're still useful, but not as useful as you wish them they would be. So if you have a combination of both, that definitely helps. Yeah, so just spend a lot of time there to make a very strong preclinical package, be very rigorous, and carefully look also in the literature what other people are getting in terms of immune responses and efficacy, so you know where the bar is that you have to cross.
Thank you. Natalia, I see that you just wanted to quickly comment on what Jose, I believe his name is Jose Luna, not Jose Sanchez. I stand corrected, apologies. Just to respond to his question, his initial one, which was virus, I believe, is that viruses against cancer sounds compelling. If they mutate, they could attack healthy cells too. What measures are there to prevent such viruses going rogue?
Yeah, generally these questions are really interesting, and I would say that viruses which are programmed to attack cancer cells usually face another problem, and this problem is quite opposite to the one described here. Basically, when oncolytic viruses are injected into the human body, the immune response develops too drastically, which means that human body produces too many neutralizing antibodies, and these neutralizing antibodies deactivate oncolytic viruses too fast. That is why generally the action time of these oncolytic viruses is too short, and basically researchers are trying to make it longer and at the same time of optimal efficacy. That is why no, I wouldn't say that oncolytic viruses are likely to mutate in a way to attack healthy cells, because all the genes are deleted from their genome, which could be responsible for targeting healthy cells. But for instance, these oncolytic viruses are very likely to be deactivated by neutralizing antibodies. And the dilemma here is that since we're using the activity of immune cells of human body, we cannot really suppress the immune response. So we should keep it active, but yeah, at the same time, it's a really, really fragile balance.
Okay, thank you. Are there any final remarks from our panelists before we close today's conference?
I think there was one remark about the cost of personalized cancer vaccines. Yeah, I mean, clearly so the cost of goods are more expensive than an off-the-shelf therapy. Mm-hmm. But it's also, of course, very much dependent on the vector that you use. I think the peptides are probably going to be very expensive, maybe too prohibitively expensive, whereas DNA is something that is pretty cheap. But of course, you still have the cost associated with actually sequencing the tumors, although those costs have come down. But you still need to do the analysis and actually make the products. But I do think, you know, if you get very profound clinical responses, then you know, of the type that people have seen with CARs, for example, or the Lulux CARs, which are also very expensive to make, that there is a room, that there is a path forward. But then, of course, the responses need to be truly stellar.
Any other final remarks?
If we can go back to the question regarding CRISPR, I remember we skipped it, but yeah, I just can give a fast remark that, like, first of all, cancer cells are not likely to mutate back to healthy cells. It's pretty much impossible because their genome and epigenome are highly aberrated. And also regarding whether CRISPR could replace anti-cancer viruses, well, I would say that CRISPR can be efficiently used to fix some of the overexpression problems, for example, or some mutations, but at the same time, we should be aware that even though CRISPR is quite a precise genome editing tool, it still has lots of off-target activity. So it was discovered just like with the whole genome sequencing technology, that basically if we are using CRISPR, we are not targeting often only our target gene. That is why, because of these off-target activities, it still remains a concern, and it still requires more research before it can be actively implemented into the clinic, even though CRISPR is used for CAR-T technology, but that's a little bit different.
Thank you very much.